Hello students, now in this lecture we will be continuing our eukaryotic cell organelle part, and in this lecture we will be talking about cytoskeleton, centriole or centrosome, cilia and flagella, nucleus and microbodies. Okay, so we will talk about all this component, and first we start with the cytoskeleton. Cytoskeleton means it is an elaborate network of filamentous proteinaceous structures present in cytoplasm of cell. So these are the elaborate networks of filamentous proteinaceous structure which are present in the cytoplasm. This cytoskeleton is involved in many or various types of functions. Okay, they can provide also mechanical support, motility, maintenance of the shape of cell. All this they can do. But in this part we will be discussing types of microtubule. Okay, types of microtubule, and this cytoskeleton is of three types. Number one is microtubules. Number two, microfilaments. Number three, intermediate filaments. Microtubules, microfilament and intermediate filament. Now first we talk about microtubules. These microtubules were discovered by Robertis and Franchi. These microtubules are found in cytoplasmic matrix of all eukaryotic cell. They occur in cilia, flagella, centrioles, basal body of flagella, and in mitotic apparatus, etc. You will find this particular microtubules, but these microtubules are absent in prokaryotes. Now these microtubules are hollow, unbranched cylinders. These microtubules are hollow, unbranched cylinders, generally 25 nanometer in diameter, with hollow core of 15 nanometer, with the central hollow core of 15 nanometer, whereas this is the unbranched cylinders and 25 nanometer in diameter and 15 nanometer hollow central part is covered. So now if we talk about a structure of this microtubule, this microtubule somewhat look like this. Okay, now if we see this and we make a larger diagram of this, and then it look like this, and this has this protofilaments. And how many protofilament are there? 13 protofilament are there. Okay, so 13 protofilament are found here. In this, each protofilament is made up of alpha and beta subunit of tubulin protein. Now the assembly and disassembly of these microtubules require GTP and calcium. Okay, they require GTP and calcium. So this is the structure of microtubule. So they have a central this hollow core of 15 nanometer, and this complete diameter is 25 nanometer. Okay, so this you should remember this diameter, that complete diameter is 25 nanometer and the hollow core is of 15 nanometer. Now we talk about functions of this microtubule. So when we talk about functions of microtubule, this microtubule help in spindle and astral ray formation. Help in spindle and astral ray formation during cell division. They form the cytoskeleton. They form cytoskeleton of cilia and flagella. And in cilia and flagella, we will going to study the structure of this, where we will locate this microtubule. Third, help in generating shape, rigidity, and form of the cells. Fourth function is microtubule help in anaphasic movement of chromosome. Fifth function is they help in intracellular transport of nutrients and inorganic ions. So they help in intracellular transport of this. So within the cell, the transport of minerals and inorganic ions are also facilitated by these microtubules. The fourth point, anaphasic movement. During the anaphase, during the cell division, the chromosome move to the opposite fold due to the help of this one. And sixth point is position of future cell plate. Position of future cell plate determined by microtubule. Okay, so this is the particular functions of microtubule, and microtubule, which is an unbranched structure and help in this many things. Now after this, second cytoskeletal structure is microfilaments. These microfilaments are found in eukaryotic cell. They are found in eukaryotic cell. These microfilaments are solid, unbranched, rod-like fibrils of indefinite length. They are of indefinite length. They are mainly composed of globular protein actin. They are mainly composed of globular protein actin, but have filamentous protein myosin also. Means they are made up of this actin and myosin protein. Both are involved in this particular formation. They form extensive network in cytoplasm cell and may be associated with plasma membrane. They may be found associated with plasma membrane. So these are the structures which are solid, unbranched, rod-like filament of indefinite length, and actin and if made up of actin and myosin, then they are microfilament. So now first we talk about the particular functions. So functions, it provides support to plasma membrane. First it provide support to plasma membrane. Second, this represent contractile system of cell. Contractile system of cell, and are involved in cytoplasmic streaming, that is movement of cytoplasm, and amoeboid movement. And fourth, they also help in pseudopodia formation, because they help in amoeboid movement, so obviously they help in a pseudopodia formation. And fifth point, which is the important function, that they help in formation of cleavage furrow during cell division. So in cleavage furrow formation occur in case of cell division of animal cell, whereas cell plate formation is facilitated by the microtubules. And here for cleavage furrow in case of animal cell, the microfilament will help. So these are the functions of microfilament. And third filamentous proteinaceous structure which is found in the cell, that is intermediate filament. Now intermediate filament are non-contractile hollow filament. They are non-contractile hollow filament of acidic protein. They are involved in formation of scaffold for chromatin and form basket around nucleus. So this is the important point. They ask directly that which of the following form a basket around nucleus. So we remember intermediate filaments form a basket around nucleus. So if we see the structure of a cell, then in cell we can locate like this, that there is a nucleus. So around nucleus you will find several like this, some of the intermediate filaments, and they will form particular basket around nucleus. Apart from that, you will find some of the microtubules in cytoplasm, and apart from that, some you will find of a independent length, and they are of longer length, that are microfilament. So like this all proteinaceous structure are found within the cell, and they form a cytoskeleton part of a cell. Okay, now these three cytoskeleton are microtubule, microfilament and intermediate filament. Out of this microtubule, which consists of tubulin protein, which is a non-contractile protein, help in formation of a structure of centriole and cilia flagella. So next, cilia flagella and centriole we will talk, and in that we will talk about this microtubule arrangement as well. Okay, dear students, next topic which we start, that is centriole. Centriole are called as cell center. This centriole occur in all eukaryotic animals. It occur in all eukaryotic animals, cells, and motile lower plant cell, and motile lower plant cells. But this centriole are absent in prokaryotic cell, amoeba, yeast, gymnosperms and angiosperms. This centriole occur in pair. And known as diplosome. So diplosome consists of a pair of centriole. So when the centriole occur in pair, suppose, and these two centriole are present at right angle, be remember, when two centriole or centriole pair present in the form of diplosome, they are present at right angle. Now these two centriole are surrounded by a clear fibrous cytoplasm. They are surrounded by a clear cytoplasm. This clear cytoplasm a fibrous cytoplasm form a centrosphere or kinoplasm. It is known as centrosphere or kinoplasm, and these are centriole. Okay, this centriole and centrosphere together constitute centrosome. They together constitute centrosome, or combinedly known as what? Centrosome. Means centrosome consists of centriole and centrosphere, according to scientist named as Bowery. And this usually lies near nucleus at interphase state. Okay, they usually lie near the nucleus. Now in this structure, if we see this part from this direction, then we find that the particular, and this centriole also, if you see this direction, from this direction, if we see this centriole, then we will find a 9+0 arrangement in case of this. What? A 9+0 arrangement of microtubule. So when we see the structure of, so next we talk about structure of centriole. And when we talk about a structure of centriole, it has 9+0 arrangement of microtubule. Now 9 means 9 peripheral triplet of microtubule, and 0 means absence of microtubule. So if we draw its 9+0 arrangement, then it look like this. Centrally we have no microtubule, that is central hub, and peripheral 9 triplet are there like this. Such a way 9 particular triplets are present. They are named, labeled as A, B, C microtubule. They are labeled as A, B, C. A towards the central. Now in this, the C of this particular is joining with the A of the next microtubule. So C join with the A. And when C join with this, this is known as CA proteinaceous linker, or CA linker will join the C of one microtubule to the A of a next microtubule. Then this all A microtubule are joined with the central hub by a radial spoke, like this. And in this radial spoke, you will find a one bulging structure before this A microtubule. They are known as X thickening. They are named as X thickening. Now these two X thickening join to each other and form a Y thickening. And this Y thickening, join with the CA linker like this. And this form a whole structure. Now in this you can see there is a X thickening, Y thickening. The structure which you find at center, this one, that is known as central hub. These are peripheral triplet. There are peripheral triplet. Then we have a CA linker, and they have a. So in this structure, they have a central hub. They have peripheral triplet, CA linker, X thickening, Y thickening. X thickening are present on the structure. That is radial spokes. So this, how many radial spokes are there? Total 9 radial spokes are there, which connect the hub with the peripheral triplet. Now when we see one triplet, in this triplet you will find that it looks like this. And in this, 3 microtubule are common. 3 microtubule are common. So total 13 globular subunits are there. These are the 13 globular subunit form A. Now then 3 are common, so new 10 will be formed. Okay, same way here also new 10 will be there. Okay, and such a way C are linked. So total microtubule, here you can see in this diagram, this A have a 13 protofilament, 13 filaments, 13 globular subunit, then this is a 10, this is 10. Why? Because 3 are common. So total microtubule in triplet are 13 plus 10 plus 10. So it become 33. Okay, so 33 particular globular protein are involved in formation of one triplet. Okay, so this is the structure of centriole. Now this centriole structure is known as cartwheel structure or cartwheel model of centriole. So next we talk about functions of centriole. When we talk about functions of centriole, the function are: number 1, to polymerize microtubule for formation of spindle fiber and astral ray during mitosis and meiosis type of cell division. So astral ray formation and spindle fiber formation is carried out by this centriole. Second function, it helps in organization and development of cilia and flagella. Then third function is centriole move to periphery of cell. Centriole move to periphery of cell and function as basal body. Of cilia and flagella. Fourth, it determines the pose during cell division. Determine pose during cell division. The structure of basal body of cilia and flagella resemble with centriole. So because of this, basal body also has 9+0 arrangement of microtubule. Okay, now someone will ask that how many total microtubule are involved in this centriole structure. So they have a 9+0 arrangement. Okay, they have a 9+0 arrangement, and 9 are triplet. So 9 into 3, that is total 27 microtubules are present in the formation of a structure of one centriole. Okay, so this is about the function and structure aspect of centriole, which is a membraneless organelle. Now next we talk about a similar type of structure in case of cilia and flagella. Dear students, one thing we have not considered in this part. You remember that there is a one structure which is found in front of this C microtubule in centriole. This is the fibrous structure. This fibrous structure is known as macule or pericentriolar satellite or microtubule generator. What it is known as microtubule generator. So it helps in generation and extension of a microtubule during the astral ray and spindle formation. So that is a microtubule generation. Now next we start with the cilia and flagella. So next is cilia and flagella. So when we talk about cilia and flagella, these are the microscopic identical structure, but even then both can be distinguished. Both can be distinguished by number, size and function. If we talk about cilia and flagella, and we first see the difference between this, then we see that this flagella are longer process. They are longer in length, while the cilia are short appendage of the cytoplasm. Flagella are 1 to 4 in number. Usually they are 1 to 4 in number. But when we talk about cilia, the cilia are 300 to 40,000, 300 to 14,000 or more in number. Means they are numerous. Okay. Then flagella occur usually at anterior end. Usually occur at anterior end of the cell, while cilia may occur throughout the surface of cell. They occur throughout the surface of cell. Flagella beat independently, while the cilia tend to beat. They beat in coordinated rhythm or a manner. So if we beat independently, they are beating like this, just they are beating independently by their own. But in cilia, numerous cilia are there and they particularly show. Now suppose this is a cell, and if we talk about, then these projections are nothing but cilia. But if we talk about flagella, flagella is longer. And this flagella and cilia, if we talk about their ultrastructure, then they have a 9+2 arrangement of microtubule. Now in this 9 plus 2 arrangement of microtubule, 9 represent doublet and this doublet are present at periphery, so they are peripheral, and these 2 are singlet at center. So peripheral doublet are there and 2 singlet are there, they are found in this structure. So now we try to draw this ultra structure of cilia and flagella with 9 plus 2 arrangement of microtubule. So if we draw the structure, then here this is the structure inner to which you will find singlet. Okay, so here 2 singlet are there, this is 1 and this is 2, and this singlet are joined by some intersinglet bridge. Okay, and 9 doublet, these are the 2 singlet, but 9 peripheral doublets are there. So 9 doublets are arranged like this, and these doublets are arranged somewhat tilted. Okay, so they have tilting like this, in which the one which is present towards the inner side, that is A, and outer side is B. Okay, they have a AB. Now from A, 2 arms are raised like this, that is A arm and B arm. This is A and this is B. So like this, A microtubule give 2 arms. The lower one is A arm, and this the upper one is the B arm. From the A arm, this will join with to the B subfibril and form a AB linker. Like this, AB linker will be formed, and here this A are also associated with this centrosphere by or central sheet by this radial spoke. Such a way they are arranged. And this is a membrane bound, so over the complete structure of cilia or in a cross section you'll find this membrane. Okay, so this is the structural part. So here if we label this, we find that these are the central cingulate fibril, central cingulate fibril. Okay, then over this cingulate fibrils are covered by central sheath. Okay, then we find a structure which is somewhat disc like, that is known as transition junction. Then we find matrix here. There are interdoublet beads, that is AB linker. These are the AB linker, which are also named as interdoublet bridge. Okay, then we have an arm, that is this is the outer arm, and this is the inner arm which arise from the A subfibril. This is the flagellar sheath or membrane which is covering the flagella. Okay, then these are the two peripheral doublet. This is the doublet, peripheral doublet, and they are connected by a linker. Then this is the spoke, radial spoke, and radial spoke have a bulging structure which is named as head. This is named as head. So such a way whole the structure of this centriole is made. Okay, cilia or flagella is made. So you can see this is a complete structure which is showing 9+2 arrangement. Now in this structure, if we note down some of the points, then we find that all microtubule are made up of tubulin protein. Okay, they are made up of tubulin proteins. Arms of a microtubule contain dynein protein, and this dynein protein has ATPase activity. So breakdown of ATP occur when which transfer the energy. Okay, ATPase activity in which breakdown of ATP occur, hydrolysis of ATP occur, and energy is released which is directly used for the ciliar or flagellar movement work. Okay. Now interdoublet bridge are made up of nexin protein. They are made up of nexin protein. So we remember the question which importantly they ask, that is of a particularly dynein protein, and dynein protein has a ATPase activity. Which particular protein has a ATPase activity? So you know that dynein protein has a ATPase activity. Now question which we can address here, that how many microtubule are present in cilia or flagella structure, and that is in the filament. So if they ask, this is having a 9+2 arrangement, these are doublet and these are singlet. So 9 into 2, that is 18, and plus 2 singlet, so total 20 microtubule are there in case of the cilia and flagella. Whereas in case of centriole we have studied total 27 microtubule. Now next we talk about functions of this cilia and flagella. Then both act as organ of locomotion. Both act as an organ of locomotion. Cilia help in aeration, feeding, and circulation of food also. Cilia of respiratory tract help in retaining dust particle and prevent entry into lungs. So it prevent entry of some dust particles in the lung that is carried out by the cilia. Okay. So till now we have discussed about the structure of cilia and flagella. Now we start with the next organelle, and which is one of the important one, that is a nucleus. We know that as our brain is important for our whole body, same way nucleus is a brain of the cell, and it is a particularly that much important for the cell. So nucleus was discovered by Robert Brown in the root cells of orchid plant. Okay, and one another scientist whose name is J. Hammerling, in 1953 he conducted some experiments on single-celled green alga acetabularia and proved hereditary role of nucleus. Okay, now if we talk about this nucleus controls cellular metabolism, encloses all genetic information and able to transmit the same through the next generation. Okay, so we are going to talk about a structure. Before that, we should talk about some of the points which are helping them to understand better. So this is also called as a master organelle. Why? Because it regulates whole particular functions of the cell, and it is present in all eukaryotic cells. It is present in all eukaryotic cell. Now some cells contain nucleus when they are young but degenerate and when cell mature. So at maturity, nucleus degenerate and become absent in mature RBC of mammals and seed tube in plants. In prokaryotic cell, true nucleus is absent, that we know very well. Generally, there is a one nucleus in each cell, but this number may vary. So depending on the number of nuclei, they can be called as uni, bi or multinucleated. So if there is a single nucleus, then there is a uninucleated, two are there, then binucleate, and three are there, then multinucleate. Now multiple nuclear condition arise either by the fusion of many cells or by the repeated nuclear division. Okay, So multinucleate condition can be termed as syncytium or cyanocytic. Syncytium term is used when multinucleate condition arise due to fusion of many cells, but cyanocytic that is due to repeated nuclear division without cytokinesis. So nucleus karyokinesis when not followed by the cytokinesis, cyanocytic condition will occur. Now we talk about structure of nucleus. Now nucleus is composed of number 1 nuclear envelope, second nucleoplasm, third nucleolus, and fourth chromatin material. We will discuss these four things in case of nucleus. So now we start with the first, nuclear envelope. When we talk about nuclear envelope, nuclear membrane is made up of two layer or two membrane, that is outer nuclear membrane and inner nuclear membrane. Outer nuclear membrane and inner nuclear membrane, these two type of nuclear membrane will be formed. Okay, so that is what a nuclear envelope. So if we draw the structure of this, we find a diagram like this. So here this is outer membrane of nucleus and this is the inner membrane of nucleus. This is nuclear pore. Then the space which is present between these two, that is known as perinuclear space. That is what perinuclear space. Then the region immediately just present inner to the nuclear membrane, it is known as fibrous lamina. It is known as fibrous lamina, and in this chromatin material is there. Apart from the chromatin, you will find one membraneless structure that is known as nucleolus, and this is the nucleoplasm. Okay, so this all things are present in case of this. First we are talking now in detail with the nuclear envelope, and nuclear membrane is made up of outer and inner nuclear membrane. Next is the space between two nuclear membrane is known as perinuclear space. Then nuclear pore is formed by fusion of both membrane of nucleus. Nuclear pore allows RNA and protein in both direction. That allow RNA and protein in both direction. Okay, so this is the nuclear pore. The sometimes outer nuclear membrane have ribosome association. So ribosome are found associated with the outer nuclear membrane. So this is the nuclear envelope. Now after nuclear envelope, second is nucleoplasm. This nucleoplasm is a transparent semi-fluid granular substance known as nuclear sap or nucleoplasm or karyolin. This nucleoplasm mainly made of nucleic acid, proteins, enzyme, lipid and minerals. Nucleic acids are DNA and RNA. They are found here in association with some proteins. Next, third is chromatin. Now chromatin is a hereditary part of nucleus. It is hereditary material or part of nucleus. It is uncondensed network of filament or fine threads. This chromatin is made up of, this is the important question that you usually find, it is made up of DNA, RNA, histone and non-histone protein. It is made up of histone and non-histone protein. Okay, so be remember, it is the important question they ask usually, that chromatin is made up of DNA, RNA, histone and non-histone proteins. Now this chromatin get condensed and differentiated into two regions. So chromatin is of two types, that is euchromatin and heterochromatin. Euchromatin is loosely coiled chromatin and heterochromatin is tightly coiled. So this is genetically or transcriptionally active. Why? Because euchromatin is loosely coiled, so enzymes can interact with this, but heterochromatin is tightly coiled, so it is genetically or transcriptionally inactive. So it is a transcriptionally inactive, then we call it as a what? Heterochromatin. And fourth part is nucleolus. Now nucleolus is an organ first discovered by Fontana and term given by Bowman. It is a spherical dense, it is a spherical dense colloidal acidophilic body. Okay, now it is membraneless, and associated membraneless, and if we talk about function, then this is involved in rRNA, that is ribosomal RNA synthesis. Okay, so this is the structure. Now one more important part, so till now we have structure with this nucleus. One more important part that we are discussing here, that chromatin get condensed and form a chromosome. Okay, and in chromosome we know that we have centromere, and surrounding to that we have a disc like structure that is known as kinetochore. On lateral side of centromere you will find a disc like structure that is known as kinetochore, and this centromere is known as primary constriction, whereas secondary constriction occur, which is known as NOR, nucleolar organizer region, and due to which the DNA gets separated here, and that is known as satellite DNA. So NOR is a secondary constriction, and NOR is nothing but nucleolar organizer region, means it helps in organization of nucleolus. So nucleolus is found associated with the NOR. Okay, and that is nothing but a secondary constriction. So that is all about the nucleus. Now we talk about the types of chromosome based on the position of centromere. Next is types of chromosome based on position of centromere. Okay, based on position of centromere. Number one is metacentric, means centromere is at middle position. So if we see, this is the chromosome, and if the size of both arm is same, and centromere is present in the middle, then it is known as metacentric. Okay, and these are V-shaped chromosome during the anaphase. Second is submetacentric, when centromere is slightly away or displaced from the center. So it lead to form one short and one long arm. You can see in this diagram, this is a long arm and this is a short arm. So like this, if we occur, then it is a submetacentric, and these are L-shaped during the anaphase. Third is acrocentric. Acrocentric means here centromere is subterminal, means near to the terminal position. So here you can see this is a suppose very long, so it leads to the very long and very short arm, and these are J-shaped chromosome during the anaphase. Fourth is telocentric chromosome, and in telocentric, centromere is at terminal position. So if centromere is at terminal position, then only single arm is found. Then these are I-shaped chromosome during anaphase. So if we see the diagram of the anaphase, it will look like this, that here centromere is there, centriole is there, like this. And if we make a proper diagram with the shape in anaphase, then if there is this arm is of equal size, then it is a V shape. If there is one is a long, one is a short, long, one is a long arm and one is a short arm, it is L shaped. Then one is a short arm and one is very short and one is very long, that is a J shaped. And when only this is present, then it is a L shaped. You can clear this is a V, L, J and I. Like this shape appear during the anaphase. So in this diagram you can dictate it. Okay. So these are the all 4 types of chromosome based on the position of centromere. There are presence of some large sized chromosome, and they are number 1 Lambrusche chromosome, and second Polytiny or salivary gland chromosome. Okay, so that is all about the part of this. Now next, after nucleus, we talk about the last topic, that is microbodies. Last topic, that is microbodies. Now microbodies are the single membrane bound small refractile bodies. These possess some respiratory enzymes other than aerobic respiration. Okay, and these microbodies are of 3 types. Be remember present in both plants and animals. These are of 3 types. Number 1 glyoxysome. This glyoxysome carry out gluconeogenesis genesis or glyoxylate cycle, where non-carbohydrate are converted into carbohydrate by glyoxylate cycle. Animals do not execute this, as they lack enzymes required for this. Okay. Second microbodies is spirosomes, and these spirosomes carry out storage of oil and lipids. Okay, so they are found in oily seeds. And third is peroxisome. Now peroxisome are involved in photorespiration, and it has two enzymes. One is glycolate oxidase, that is marker enzyme, and second catalase. Okay, so these two are enzymes are present, and this peroxisome perform photorespiration by association with chloroplast and mitochondria. And this photorespiration occur in C3 plants that we will study in the photosynthesis chapter. Okay, so that is about what a microbode is, and that is all about the chapter cell, the phenotype life. I hope you understood this. So please go through this each and every time. Wherever you find difficulty, go again and again, and you will understand. We will meet in our next chapter. Thank you.